Good morning, everyone. We will continue where we left yesterday. In the last class, we have discussed about a nice vector, pGem, which has two promoters, not just one. And we discussed how they can be used. They can be expressed. One of it used can express one gene in one orientation, and if you happen to clone it in different orientation, you can express it through sp6. sp6 and t7 are promoters derived from viruses, and these viruses have their respective polymerases, t7 polymerase or sp6 polymerase, and they have to be expressed from outside. So you can synthesize either of the sense strand or the ANSI strand based on the orientation of the open reading frame that is cloned. There are also other uses for using such two promoters. Say, for example, I had an open reading frame cloned in this direction. This is the top one, okay? And I want to make a probe for it, right? Probe would have been the bottom strand or antisense of it. So I could have used sp6 promoter to synthesize the antisense of this for this mRNA, and then I can use it for northern blotting. Okay. And we also discussed about yeast episomal vector. Episome is something that stays independent of the genome. That means it doesn't get integrated into the genome. And we also discussed about what makes something shuttle vector. It depends on the hosts or organisms that this particular vector is going to be used in. Say, we would do all the manipulations in E. coli and then probably do something else with the yeast. So for this purpose, we need two origin of replications, one for E. coli and one for yeast. And then we also need two selectable markers, one in E. coli and one in the yeast. So here are origin of replication for E. coli, origin of replication for yeast, selectable marker for E. coli, and selectable marker for TRP1. Regarding TRP1, we discussed about what is meant by an auxotrope and what is meant by a prototrope. Prototrope is the wild type, and auxotrope is a mutant, which is deficient in one or more of genes, and it cannot grow on minimal media. It should be supplemented with whatever is missing. Say in the example yesterday we discussed, we are in the yeast episomal vector that is depicted here, we were talking about tryptophan. That means this particular cell cannot grow on minimal media unless it is supplemented, the minimal media is supplemented with tryptophan. Pre-synthesized exogenous tryptophan should be supplied, which will be taken up and then used for protein synthesis. We also discussed a few things about we discussed about another one, which is referred to as the yeast centromere vector, because in the previous one, we don't know if it will be inherited faithfully to the daughter cells, right? Because it's an episome. If we want it stable and effective or efficient inheritance to the daughter cells, we should have added centromere. The centromere will help the synchronization of centromere plus autonomously replicating sequence. These two will synchronize the replication and segregation of this vector into the daughter cells along with that of the genome, because of centromere is where the kinetochore forms, and the spindle fibers will pull them together to each pole of the cell, thereby each daughter cell getting one copy each. And the cell cycle in E. coli is much more complex, isn't it, compared to that of bacteria. So we have G phase, G1 phase, S phase, G2 phase, M phase, and then the cycle goes on. Replication, um, pre-initiation or replication or initiation happens some other time, and activation happens at a different time. So if we want this to be completely synchronized, because the copy number should also be maintained, we want the the, the yeast vector here to be synthesized along with that of genome, then we should add autonomously replicating sequence. Thereby, the daughter cells will get one copy each. So when you're reading, please look at the legend, and everything is here. And I'm trying to explain more of those things. And today we will discuss a little more on yeast integrative vector. So why I'm going into depth of yeast based ones is it's eukaryotic, and mammalian and other ones would also be similar. And these are the varieties. In the first one, we saw it as an episomal vector. In the second one, it is centromere based vector. And now we are looking at something that can go integrate into the genome itself. So first, as usual, we should always look into what are the things that we have. We have EcoR1 for origin of replication for EcoR1, beta lactamase for selection against antibiotic resistance. And then we have, this is a recombinant which has the gene of interest. And now there is something else, URA3. So the cloning sequence or the selection scheme for it is what we are trying to understand better. Okay. So we are having URA3, which is say utilized or useful in uracil metabolism or synthesis. Okay, uracil is important for in as a part of RNA, right? RNA has GAUC, uracil instead of thymine. So if we are looking at, this is a functional gene, uracil. And what you're showing here is chromosomal DNA has a mutation in uracil 3. That means you can think of the host that is taken here, yeast, is oxotropic for uracil. What do you understand by that? That means this particular host that we are going to use is deficient in synthesizing uracil. So if we want this particular yeast mutant oxotrop to grow, we can grow it in rich medium, because rich medium may have uracil already, or more minimal media plus uracil. If you have, it cannot grow on, I'll put it like this, it cannot grow on minimal media, but it can grow on minimal media supplemented with uracil. Okay. So this is what we are going to use, minimal media for selection scheme. There is another aspect to this, that it is about homologous recombination. So here is the uracil gene, but there is a mutation because of which this yeast has become oxotropic to uracil. It cannot synthesize uracil by itself. And I hope you also know homologous recombination. In the first beginning of this course, I made you do some assignments with homologous recombination as well. Homologous recombination that happens between homologous sequences. When we say homologous sequences, that means they have a lot of similarity, sequence similarity between these two. There is a small difference here as such as well. So if I take the host and I grow it in minimal media, it will not grow. If I take host in minimal media plus uracil, then it will grow. Now, how we are going to employ this is if recombination, when we introduce the host, say, for example, the host is here, and we have introduced the vector. We will call it as Y, yeah, we will call it as YIP. But it has not integrated. Say it has not yet integrated. Will it grow or not on minimal media? It will not grow, mostly because although the cell has the YIP but did not get integrated, there is no promoter for it. Okay, we will not have efficient expression of it. And there is one more thing that you also have to remember is there is no origin of replication for yeast. We did not put that in this vector. So if there is any way that the yeast integrative plasmid is going to replicate, it is only if it gets integrated into the host genome, because genome has ARS, autonomously replicating sequences, and therefore it will replicate. And if the vector that we are introducing gets recombined with the host genome, then it will also get replicated along with the genome. Otherwise, there is no way that this is going to replicate. So if we have a host and YIP is integrated, then it can grow on minimal media, because now there is a functional uracil gene that has formed upon recombination between these two. We have a functional uracil form, and it will be expressed, and it will produce the enzyme required for uracil synthesis, biosynthesis, and then these will grow. Okay, so let's just take one simple way of how we will see the scheme itself. So we will take YIP and we will add yeast cells which are oxotropic to uracil minus, and then we will get 2 or 3 different types of cells. One of it is transformants, or maybe you can say it as transfectants. Non-transformants, and we'll also get transformants. And in the transformants, it might get integrated, right? If it is not integrated, because there is no origin of replication to replicate in the yeast, it will not replicate any further. And it is, I described you how important is an origin of replication. All genomes, all propagating entities have one thing, and that is the origin of replication. If it is not there, it is as well just a piece of DNA. If origin of replication is present, then it becomes something different, propagating entity all of a sudden. Okay, the transformants will get integrated. I mean, within the transformants, you'll find the integration of the YIP, and then what you will grow it on is you will put them, excuse me, we will grow both of these cells in minimal media. That means we haven't supplied any uracil. So the non-transformants, because they are uracil minus, they will fail to grow, whereas these will grow. okay, because they have formed a functional uracil gene. That is how yeast integrative plasmid works. So I hope you're trying to follow the scheme or where to focus. Everything is in the DNA. You should know why some things are there and why some things are not there. You want to force, or you want only the ones that are integrated. That means you should not have put any origin of replication for uracil. If you put, it is as well as the previous one or the centromeric one or the episomal one, right? I hope you understood this. So there are several other similar expression vectors for mammalian cells. I'm not going to go into deep, but I hope you will be able to understand. This has to grow. It is an episomal vector. I hope you remember what is an episome. Episome is a replicating system that is not integrated into the host genome, but it exists in the cell. So we have a cytomegalovirus promoter that is here. This is the promoter. Here is the multiple cloning site where you are going to clone the gene of interest. And here we have PA, that is polyadenylation signal. I may have already described, but I hope you know the properties of eukaryotic and prokaryotic mRNAs. They structurally or chemically, how are they different? In eukaryotes, you have a G cap on the 5' end and a poly A tail. This G cap is important or essential in most cases for the translational initiation. If it is not there, it doesn't initiate. Translational initiation may not happen properly. And you also have the poly A tail, which is a kind of, it will allow the circularization of, at least by one model, it allows the circularization of the mRNA with the G cap like this, and translation keeps on happening. Right? When the ribosome comes here, it will dissociate and reform, start initiation here again. So basically, the two functions of poly A tail and the cap are stability of the mRNA, because if they are not there, usually the mRNAs are degraded. And secondly is that they increase the efficiency with which the mRNA is translated. Both ways, it will contribute to the increase of the amount of protein that is produced. So we know about EcoR1 origin of replication, beta lactamase. So cytomegalovirus, a promoter from there, which is a strong promoter, which will express whatever gene we may have cloned in the multiple cloning site. And the mRNA should end with a polyadenylation signal. So we have put in there as well. And we want this vector to grow also in mammalian cells. So we have taken simian virus 40 origin of replication. And it will remain in the nucleus. Okay, in the nucleus, it is not integrated into the genome, and it will exist as an episome like that. That is an episome. Excuse me. Okay. Yes. So is there anything else? Okay, there is one more. Selection markers. For E. coli, we have beta lactamase. In the other one, we have neomycin resistance gene. Neomycin resistance gene. Neomycin resistance gene is usually a phosphotransferase. It's an enzyme. Neomycin is an aminoglycoside antibiotic, which will interfere with translation. You can find out the molecular mechanisms. So this enzyme, as the name suggests, it will phosphorylate neomycin, because of which neomycin will not grow. Okay, sorry, the cell will grow, and neomycin is no longer effective. So that is how the resistance mechanism works. So typically it would have been something like this. You would have taken some mammalian cells and you would transfect the simian virus, the episomal vector. So assume this as, we'll call it as episomal vector for mammalian episomal vector. And then we will get two, there are transfectants and there are non-transfectants. So, and then we will grow in media, culture media, supplemented with neomycin. The transfectants will survive because they have the episomal vector in which neomycin phosphotransferase is present, which will deactivate or inactivate neomycin. So there is growth. In the non-transfectants will not grow because the cells are still sensitive to neomycin. Okay? Yes. So maybe I will take one of these and let's see what we have in here. So this is a retroviral vector. I hope you know what is meant by retrovirus. Those who have studied microbiology should have, they are proficient in that. We have DNA viruses and we have RNA viruses, single-stranded, double-stranded, and so on. And in the RNA viruses, in which there is an intermediate form of DNA form, okay, they are normally packaged, when they are packaged as viruses, they have RNA in them. But after infection and during replication somewhere, if they have a DNA form, then this is backwards, right? From typically we get DNA, from DNA we get RNA, but from RNA we are getting DNA, so you call it as retro. So retroviruses are those that have an intermediate form of DNA. So here we are going to see how this can be used as a vector, and this has its own purposes, of course. Every majority of the viruses, they will have ends. Okay, if this is the linear structure of the architecture of a virus, they have long terminal repeats. This would be called as 5' LTR, long terminal repeat, and this is the 3' LTR. So when it is expressed, what we want is that it forms, if this one is in RNA, and it will produce DNA, and that DNA could get integrated into the genome. Okay, so that is how the retroviral vectors can actually be useful. Cis-acting, essential cis-acting retroviruses, PSI. This is neomycin phosphotransferase. It will also be sometimes referred to as NPT. I think this is much better to use NPT instead of neo. But yes, cytomegalovirus promoter, we already discussed about it. We have a multiple cloning site, which is now already with insert. And then we have the 3' LTR. Anything between this, the 5' LTR and 3' LTR are the ones that can get integrated. This is the viral part. The rest of the part from here to here, it is only for cloning purposes. Origin of replication in E. coli and beta-lactamase or ampicillin resistance gene. So yes, these are the things. What they have, if I put it in PSI is probably, I'm unsure. I'll try to find out if there is any specific significance here. We have neomycin or resistance gene. We have a CMV promoter, and this is where we are cloning the gene of interest. And so it will form a double-stranded DNA, and that could be used to get integrated into the genome. Okay. that is about retroviral vectors. So now we are increasing the goal of all these vectors. Have it is in special cases there are needs, like we want to have a special condition integration or a zipisome and all those things, but majority of the times it's also about the amount of fragment that could be cloned, the size of the fragment. So if we take plasmids or cosmids even, or phage based ones, there is a limitation. What if we want to not clone about kbs, but we want to clone very larger sizes? Then we need to have, usually in theory, you can also construct a plasmid very huge, right? Even if we construct it as a cosmid and put it into the bacterium, there are stability issues. Somehow these large fragments will undergo recombinations and many problems, because of which their stability is a problem. So stability in the sense, as a structure itself, there is stability, okay, and whether it is going to be inherited by the daughter cells or not is another problem. So we want to have large fragment. We want to have a large fragment cloned. We want increased or no compromise on stability. We also want the vector to be inherited faithfully to the daughter cells. And in that case, here is a yeast artificial chromosome, YAC, yeast artificial chromosome, and it has several factors. And every chromosome requires three important parts. First one, of course, is the centromere, like we discussed before. There are also the telomeres, especially if they are in linear, we would need telomeres. We also need ARS, right? The origin of replication sequence, origin of replications. This is what we need to have for a DNA to be like a linear chromosome. This is exactly what they have. So from here, crossing, I mean, all this is a chromosome. From BAM-H1 to the BAM-H1 is a chromosome. Here we have just a stuffer region. So there is a centromere. So we got centromere. We have telomeres, that is also there, and we have ARS, that is for origin of replication, that is also there. What we also would need is TRP for selection and URA3 for integration, or this is also another selection. Okay, tryptophan gene for biosynthetic gene, as well as uracil, they can be used as selectable markers. So what they would have done is cut it with SNAB1 and BAMH1. BAMH1 would cut here and here, and SNAB1 would cut here. So we are getting three different fragments. The stuffer is, of course, discarded. Then we are left with left arm, that is from here, starting from BAMH1 site till the SNAB1. That is the left arm. The right arm is from SNAB1 to BAMH1. That is this right arm. Now they will ligate a large fragment of DNA, and then which would have for SNAB1 and a very large fragment, and the DNA, the chromosome would be something like this. Of course, there are other possibilities as well. So here the host that we choose. Say this is yeast, right? The yeast host that we are choosing, or yeast auxotrope we have chosen, which is trip minus, it cannot synthesize tryptophan, and uracil minus, it cannot synthesize uracil as well. So when I said we have ligation to do, what are all the possibilities? This is the left arm, this is the insert, and this is right arm. LIR. There are also other possibilities. LIL or III. It can be multimers of it. You can also have LR, isn't it? Because we used SNAB1. Maybe we could include a step of we could also include a step where we might have treated these two with alkaline phosphatase so that self-ligation would not have occurred. So what else is possible? RIR, right arm, insert, right arm. So which ones will grow, which ones will not grow? Can anybody try telling me? This is the scheme that we have. This is the host. So the left arm, what is the first? Left arm has trip1. Right arm has uracil.
Uracil.
That's right. So which of these combinations is likely to grow on a host like this? And you are plating them on minimal media.
Which
Will
contains both,
LIR grow? Yes, yes, please.
which contains both the left and the right arms, sir.
That's right.
That
So
LIR, sir.
LIR will grow. This will not grow LIL because it is missing the uracil gene. III will not grow because it will miss both. This will not grow. This is also likely to grow. Unless if we have treated with alkaline phosphatase, this may not have formed. Right?
Yes, sir.
If yes.
Yes, sir.
So that is the scheme for how or the design. Everything is in the vector. All we need to know is a little bit of biology that is associated with each terms. Why do we have it in the first place? Why do chromosomes have telomeres? Telomeres are important for what is the importance of telomeres? Anybody? Yes, please. Somebody can try. Don't worry if the answers are wrong. That's all right. No issues.
The DNA from damage,
DNA?
from the damage, sir, like during
Oh,
replication.
what is that called as? What
End
is end
replication
replication
problem.
problem? Yes. With increasing number of, after each round of DNA replication, please have a look at it. I wouldn't have enough time to discuss. There will be a primer synthesized, and then that means we will not have any way of synthesizing the last bit of the 5 prime bit of the DNA. So after each round of replication, there is a possibility that the chromosome becomes smaller and smaller. So telomerase is the enzyme that makes a big addition here and here. So even though the cell undergoes, it is reducing not in the essential genes, but in the telomere size is going smaller and smaller. But at one point, that is one of the reasons why cells have certain a certain number of population doublings. And unless like in embryonic stem cells or in the case of cancer cells where telomerase is highly expressed, they can divide multiple or unlimited number of times. And there are also other issues. Telomeres also form a knot-like structure at the end and then they are coated with some proteins, which otherwise, if DNA is like this, the ends are vulnerable to exonuclease activity, right? If it is circular DNA, no issues. It is like this, no problems. No ends are freely accessible by exonucleases. But in linear DNA, there are problems. So telomeres also form some structures like this, which make them which prevent them from activity by exonucleases. Degradation is reduced. There is also one more is that, let me erase. Say in the DNA, in the genome, we have several chromosomes, we have about 23 pairs of chromosomes, right? And we also have repair mechanisms, something like double strand break repair mechanisms. Say this is chromosome 1 and this is chromosome 2. What will prevent these two from being repaired by double strand breaks or non-homologous end joining to make it as a fused chromosome like this? Because we have telomeres, that will prevent them from forming, that will prevent this recombination of these chromosomes. Okay, so that is why we need to have telomeres, and we should know. And of course, in evolution, if you see, some of the organisms do have fused chromosomes, and that, I think, that is one of the difference between chimpanzees or about the specific details. At the same time, we should have known what is the significance of centromere. Why is it important, or what does it do? It is essential for segregation of the chromosome to the daughter cells. And how is that? Because that is where the kinetochore would have formed. And then when you have two sister chromatids, each one is pulled towards the opposite poles. And you should have known the ARS, or origin of replications, and importantly also the TRP and uracil. In this case, you should also have known why is TRP here and uracil here, right? Everything is in the design, and that is where the intelligence comes here. They put it that way so that it becomes logical during selection, because it is on one left arm has one selectable marker, and right arm has another selectable marker. Okay?
Yes, sir.
That is about, yes, yeast artificial chromosome. Similarly, there will be mammalian artificial chromosome as well, I think, or bacterial artificial chromosome was also made. This is phage artificial chromosome. What I want you to do is try to know, you have to find out there are tables that you can see, even Wikipedia has some things. And when questions come in CAA or in the final exam, an objective can be given. I have an yeast cell, I want to, or I might not even say that is an yeast cell. I want a fungal gene expressed, or I would even change it even further. I'll simply say, I want to characterize a fungal gene, fungal protein, but I don't have the protein. I only have the gene for it. Then I might ask you to describe the cloning and purification. I might ask you to design the vector, choose a host, and also make any modifications or manipulations that are required to get the fungal protein. So everything is in the open. It's not necessary that everybody get the same. It's not one answer, it is multiple answers. At the end, it is about how accurately and cautiously you are thinking about. When you think about it, you have to look for two things. I would say speed and accuracy. That is what is going to make it efficiency, you can say. And then it should be about within the constraints. say if I have added that I only have these proteins and I don't have anything else, what should I do? Right? So try to think on those lines. Please go through over it and try to practice a few more. Practice how you would do. You can also have it as a discussion among yourselves. If I said fungal protein expression, all I have is only the gene, and gene may have introns. So I cannot use E. coli as a host for expression of it. I should have chosen yeast in that case. If it is yeast, then I should have chosen the vector accordingly, which is, but manipulations may be happening in E. coli and expression would be happening in yeast. So the promoter should be active in yeast. There should be a replication of origin for it and as well as within E. coli. You should have known how you're going to select for transformants in this case and transfectants also in the yeast. How you are going to differentiate in E. coli itself, you will differentiate which one is a recombinant and which one is not a recombinant. And you have to design the whole things. If you have performed one of it like this, the rest of the things are all easy for you, and then you can actually feel like recombinant DNA technology is done. You're fine with it. Okay, so it's important to know what all tools are there. It's first important to be aware of the biology first, the fundamentals of cell biology and molecular biology, upon which rDNA is built. Without strong foundations in biology, rDNA becomes little superficial. Anyway, we have discussed about plasmids, phage vectors, cosmids. I didn't discuss what is meant by plasmid or phagemid. I didn't discuss pack. We discussed these two. So I don't need that you discuss anything about pack, but I want you to know what are these two. What is a plasmid? What is a phagemid? That is your homework. And also try to you can each give one question to each other and try to have a discussion on it. And this is something I have an assignment for you. This is called lambda zap vectors. You may already be aware of it. I will likely give it as a CIA question. It is self-study. And if there is anything, I will discuss only after the CIA. Okay? After the exam is done. Why I do those? That is, if we have understood all these things that we have discussed so far, and the same things done a little bit differently is lambda zap vectors. First thing is try to know the design of it, architecture. What are presents in this? What genetic elements are there? And what genetic elements are there? Why are they included? What is their significance? How are they going to help achieve the goal? And then that is where you will know the features and their significance. And then you will know what all it can be used for, lambda zap vectors. Okay, that concludes the lesson on vectors. So what I want you to understand is there is no textbook. There is textbook, but reading and mugging up is insufficient for this course. If you remember the learning outcomes of this, it is an advanced course. It is in your final year, so where you are supposed to apply the biology that you have learned so far to achieve your biotechnological goals, right? Like protein production, protein characterization, and so on. So I want to discuss one thing right now is there will be a scientific communication written assignment. I want to describe it a little bit. Written assignment. If anybody is interested, or it is actually a compulsory one, you can choose any topic which is around rDNA, or maybe also regenerative medicine. So if I didn't, if I told you about the significance of rDNA, this is, say, biotech. The whole of it is representing biotechnology. Okay, if you remove rDNA, it's not biotechnology. That means why we are giving it so much attention is we are able to manipulate the genome or the genes or genetic elements to get what we want. This is all the basic science, or from um, the knowledge about biology, molecular biology, cell biology, and all those things. And here is the application. We want a large amounts of a protein, or we want to make a vaccine. We want to have gene therapy. Anything we do, the center is rDNA. Okay. So I want you to find out any one topic that you are interested in, highly specific topic, and then which will also include rDNA. If it is biotechnology, at least if you say, I want to produce this particular protein in large quantities because it's useful somewhere, you would have done it through rDNA. Somebody would have done that. So take a topic that has rDNA and write about 4,000 words, right? There should not be any plagiarism, not allowed. You should have had at least 30 references in it, citations, citing at least 30 articles. And it will be nice if you could make at least one diagram. Okay, this is like trying to give you an opportunity to write a paper. As long as you meet these, as long as you meet these conditions, you will get full marks for the assignment. How great, how good, that is all different. There should not be any plagiarism. That's not allowed at all. Okay, and you should have written at least 4,000 words, at least. You could have a 5,000 or 6,000, but minimum is 4,000. And there should have been at least 30 references, and make one diagram by yourselves. Try your best to do those things. And I will post an assignment in your GCR. What I want you to understand is, from this exercise, you will know how the publication work, the process of the whole writing. Writing is difficult, reading is difficult, and writing is difficult. For us, if you see, playing is easy, right? Singing is also kind of easy. At least anybody can do, other than the perceived humiliation. You can speak, that is also easy. Maybe not in front of people, but you can do it. What is not, because these are all natural things. All monkeys also do it, dogs also do that. How humans are different is with reading. We are different in writing. Reading is difficult, and writing is even more difficult because they are not natural or inbuilt in our system. Humans have invented it, right, in the form of a language. So these two are difficult skills for anybody, and we are supposed to, if one wants to be a successful scientist, it is important that we read more and write more. Okay. And once you start doing this, I mean, reading a novel is still good good. I appreciate that reading novel is also nice, but reading science to not it's not about just it doesn't appeal to just our fun thing or so. It is about knowledge acquisition and intelligence where your brain may have trouble. And if one is engaged in that for a long time, and writing becomes more, because if somebody is reading say 30 papers and trying to synthesize new sentences, trying to integrate all the information from them, then that's even more harder. So go through the process. Don't worry about validity. Don't have any typos or spelling mistakes. You can use Grammarly to correct all those things. Go through the process, and if everything works out, we can even try to submit it for publication. Don't think it is too theoretical. I actually tried it. I'm still trying it. What was an assignment, some not during the COVID time, but before, has is submitted for a review, is submitted for publication in a journal. So if it is rDNA based, and don't mix it up with your project works, because there will be conflicts again. I don't want that. You can take even the project work also. If you want to take it, you have to discuss with your guide and say that you're going to submit as an assignment. And then we can, if we are going to get to this submission level, then we will discuss more. Any topic that has rDNA, you can write. If you're familiar with it, it's fine. Do you have any questions on these? Anybody?
Is there a specific deadline before which we have to submit this?
Initially, I would have a deadline for this part to finish. For this process to happen, okay, usually it takes a long time. You will need to go through several refinements and see if it is still publishable. And if it is, for this is timeless. It is endless. We keep trying. So for the assignment submission, the deadline would be that you make up these things. For submission, we have to take it to a professional level, right? This is the basic. Once you have done that, and if you are interested, we can go to this level. I will tell you a deadline for this. For the basic part, we'll have a deadline. I'll discuss with you people and decide. Okay?
Yes, sir. Thank you, sir.
Yes. Take care and have fun.
Thank you, sir.
Thank you very much. I appreciate you people coming and attending regularly. Yes, thank you very much.
Thank you, sir.
Thank you.